Let's see how microbes can cause disease. In order for any pathogenic microbes to cause a disease, it has to follow these steps. The first step is the microbe should be able to gain access to the host via any of these portals. Once it enters the host, it should have some virulence factor to adhere to the host tissue, and it should have a specific number to cause a disease. After adherence, the microbe should be able to penetrate into the deeper tissue. At the same time, should be able to evade the host defense mechanism or host immune system. Once it has established itself or colonized, it should be able to cause damage to the host directly or indirectly. Finally, the microbe should be able to exit the host to infect other persons in order to spread the disease. Let's discuss all of them in detail. Microbe can enter a human host through three main portals, that is mucosal membrane, skin, or parenteral. Mucosal membrane is present in gastrointestinal tract, respiratory tract, urinary tract, and conjunctiva. Respiratory tract is the most easiest and frequently used entry site for microbes, and these diseases like tuberculosis, whooping cough, influenza, and measles. Gastrointestinal tract is also a common site for entry for many microbes, leading to diseases such as cholera, typhoid, hepatitis A, and mumps. Genitourinary tract is the entry site for most of the sexually transmitted diseases like herpes virus, AIDS. Most of the eye infections are caused by entry through the conjunctiva, which is a membrane covering the eye and the eyelids. Some microbes can gain access through hair follicles and sweat glands. Ectator americanus is a hookworm that bores through the intact skin and produces enzymes that break down keratin. In parenteral route, what happens is the microbes are deposited directly into the tissue beneath the skin or mucosal membrane via punctures, injections, bite, cut, wounds, or surgery. They may lead to diseases such as tetanus, hepatitis B, rabies, malaria. Each microbe has a preferred portal of entry, which is a prerequisite to cause of a disease. For example, Streptococcus pneumonia that are inhaled can cause pneumonia. If you swallow them generally, they will not cause any disease. The second step in disease progression is adherence, in which the microbe should be able to attach to the cells present at the site of entry. So this attachment between the microbes to the host tissue requires specific molecules called ligands or adhesins, which are present on the surface of the pathogens. These ligands or adhesins can be present on glycocalyx, flagella, fimbriae, or viral capsid. They attach to specific molecules present on the host cell surface called receptors. The second type of adherence can be through biofilms. These biofilms are communities of microbes and extracellular substances which help them adhere to both living cells and to non-living surfaces. An example is dental plaque. These biofilms also help the bacteria to evade the host immune system. They are generally resistant to phagocytosis as these phagocytes cannot enter the extracellular polymer substances of biofilm. This EPS may shield the antigen so they cannot be recognized by the immune system. Moreover, EPS of Pseudomonas aeruginosa kills the phagocytes. A microbe can enter the host cell or evade the host defenses by using different virulence factors. The first factor we discuss is capsules, which prevents the microbes from being digested by the host by a mechanism called phagocytosis. These capsules are produced by many pathogens such as Klebsiella pneumoniae, Haemophilus influenza, and Bacillus anthrax. Second type of virulence factor is present on cell wall. For example, an M protein, which is present in Streptococcus pyogenes and helps in attachment and resistance to phagocytosis. Similarly, wax is present in the cell wall of Mycobacterium tuberculosis, helps it prevent digestion after being phagocytosed. There are many different enzymes which helps in the invasion of the microbe into the deeper tissues. Let's discuss them one by one. Coagulases. These produce clot in the blood, which may wall off sites of infection from immune response. They are produced by some Staphylococcus. Bacterial kinases. They break down clot produced by the body to isolate infection. These are made by both Staphylococcus and Streptococcus bacteria. Hyaluronidase breaks down hyaluronic acid, which holds cell together in a connective tissue. These are made by Streptococci and gangrene-causing Clostridia. Collagenases break down collagen, which form connective tissue of muscle, skin, and organ. These are produced by several Clostridia. Necrotizing factor, they kill the body cells. Hypothermic factor decrease the body temperature and prevent killing of the bacteria. Lecithinase destroy the plasma membrane of the host cell and helps penetration. IgA proteases break down the IgA antibodies. Now let's have a closer look at how these virulence factors function by using two of the examples. The first is how hyaluronidase works. It's released by pathogens outside the cell, and these enzymes break down the glue which holds the two cells together, epithelial cells together. As a result, this bacteria gain access into the deeper tissue. Now let's look at how coagulase and streptokinase work. The pathogen produces coagulase to form a clot to evade the host immune system. Now see, blood clot is formed, and when the cell wants to multiply and gain access to the host bloodstream, it uses streptokinase, which dissolves the clot and releases the bacteria. Now let's see how the microbes damages the host cell. There are three basic mechanisms of damaging to the host cell. Direct damage, use of host cell nutrients, or by indirect damage by production of toxins. Some bacteria can induce cells to engulf itself, such as E. coli, Shigella, Salmonella. The cell multiply in the host cell and kills the host cell. That's called direct damage. The second type of damage is by using the host cell nutrient. Iron is required for the growth of most of the pathogenic bacteria. However, the concentration of free iron in the human body is fairly low because most of the iron is tightly bounded to iron transport protein such as lactoferrin, transferrin, hemoglobin, and ferritin. To obtain iron, most of the pathogenic microbes secrete a protein called siderophores. Siderophores are released into the medium where they take the iron away from the iron transport protein by binding to iron even more tightly. Once the iron IgG4 complex is formed, it is taken up by the bacteria or microbe. Toxins are poison substances produced by microbes and are frequently the most common pathogenic factor. Effective toxins are fever, cardiovascular problems, diarrhea, shock, destruction of red blood cells, white blood cell, and nervous system disruption. Of the 220 known bacterial toxins, 40% of them damage eukaryotic cell membrane. There are two types of toxin, exotoxin and endotoxins. We know them because we have discussed them in prokaryotic cell structure and function. Let's discuss exotoxins. Exotoxins are generally produced by gram-positive bacteria. They are produced within the bacteria and released outside. So there are various different types of exotoxins like neurotoxins which cause damage to the nervous system or nerves, even tetanus and botulinum is caused by neurotoxins. Cytotoxins are those which cause death or damage of cells. For example, in diphtheria, nerve cells, heart cells, and kidney cells are damaged or killed. Enterotoxins are the one which affects the gastrointestinal tract cells, as in case of cholera. Leukocidin destroy WBCs and produced by Staphylococcus and Streptococcus. Hemolysin destroy red blood cells produced by Clostridium perfringens. Exotoxins are antigenic. Antibodies called antitoxins produce immunity. Toxoids are toxin inactivated by either chemical or treatment and can be used to prevent infections caused by exotoxins. Endotoxins are lipid portion of LPS called lipid A. They are excreted when the cell dies and cell wall undergoes lysis. It produces signs and symptoms such as fevers, chills, and pain. These are not antigenic and do not induce immune response. Examples of bacteria which release endotoxins are Salmonella typhi, Proteus, Pseudomonas species, etc. Let's compare exotoxin and endotoxins. Exotoxins are released outside the cell, whereas endotoxins are only released when the cell undergoes lysis. It's produced mostly by gram-positive bacteria. Endotoxins are produced only by gram-negative bacteria. Exotoxin is generally present in form of subunits which combine together to give its effect. Endotoxins are basically lipid A portion of LPS. Exotoxins are relatively unstable at high temperature, that is 60 to 80. However, endotoxins are quite stable. They can withstand autoclaving, that's 121 degrees Celsius for 1 hour. Exotoxins are highly toxic as compared to endotoxins. Exotoxins don't produce fever. Endotoxins do produce fever. Exotoxins are lethal even in small amount. That's why their toxicity is high as compared to endotoxins. They should be present in a very large number to be lethal. An example of diseases caused by exotoxin is tetanus, botulinum, diphtheria, and scarlet fever. An endotoxin causing disease are typhoid fever, meningitis. Diseases caused by intake of toxin are called intoxication. and not infections. Now let's look at the different stages of disease development. Once a microbes overcomes the defensive host and develop a disease, there are the following sequence of events that takes place in case of acute disease. The first stage is called incubation period, in which there are no signs or symptoms of the disease. It is the interval between the initial exposure to the appearance of the first symptoms or signs. In some diseases, the incubation period is always the same. In others, it is quite variable. The time of incubation depends upon specific microbe, the number of infecting microbes, and the resistance of host. The second stage is called prodromal phase, in which the early signs and symptoms for disease appear, but these symptoms are vague symptoms such as general pain and malaise, and it is often difficult to differentiate between common cold from the prodromal symptoms related to other diseases like measles, chickenpox, or cytomegalovirus infection. The third phase is called the invasive phase, in which the signs and symptoms are quite severe. The disease is highest at its peak or acute, and the specific symptoms of disease appear like fever, chill, muscle pain, sensitivity to light, sore throat, lymph node enlargement, and gastrointestinal problems. In the fourth phase, that is the decline phase, the immune system gets hold of the problem and the signs and symptoms starts declining. That's called the decline phase. If the disease is not successfully overcome by the immune system or treated, the patient dies during the invasive period and never reaches the decline phase. The last phase is called Wilson's period, during which the person regains strength and the body returns to its pre-disease state. Although the person has recovered, but still the person can serve as a reservoir of the disease and can easily spread the infection. Remember, both incubation period and convalescence period can be the period in which the infection can spread from one person to another, especially in case of typhoid fever and cholera, and may last even for months or years. It's time for our last checkpoint. That's concept checkpoint number 3. So try to answer these 3 questions and we will discuss the answer in our interactive sessions.